All right, welcome back everybody. Today we're going to do some annotation for the InSituType Spatial Transcriptomics project that we're working on. So I've run everything because I realized after last time that these videos can be incredibly long. So I have run everything, and we'll just go over kind of the results as they come out. But if we go back to here, in the InSituType actual GitHub repository, they have a vignette section. So the vignette section that I have used in the past is semi-supervised, supervised, and then clustering. There's a handful of different ones. For example, if we use the semi-supervised clustering, what this one is really nice with is having the cell types that we know are in our sample. So this is a brain sample, and previously we had seen the different cell types that NanoString has annotated for us already in the metadata for the file. And if we have those gene expression profiles, as they call them, we're able to annotate our data, remember our cells, to those single cell expression profiles that we're getting from another source. So for example, in the Human Brain Atlas here, there's also an eBrain version, but they have, so if we go to eBrains, they have a data, and then if we go to, perhaps it's not as easy as I thought. So there's data, we can explore, Brain Atlas, so on and so forth, find data. We want to go and we actually look at the data that they have here. If you know exactly what you're looking for, I don't know anything about brain, nor do I know great resources for it, but you can pick things like the species that you're working with. We're working with human, it's the human frontal cortex. The brain region, you can do frontal cortex, for example. There's a spatial transcriptomics technique and all that kind of happy stuff in here. So you can just narrow it down and get the actual data that you're looking for. There's brain scans and stuff in this particular data set, so or this website. So it's a lot here, which is great if you're a brain researcher. I don't know a great deal about it though. There's also the Brain Atlas. I've tried, the reason why I'm showing these and not going too deep into it is I have tried with a expression profile. So if you go back to this guy, I do have an expression profile from, I think it's the brain, I think it's this one. Maybe it is the Brain Atlas. Anyway, I have the expression profile. Yeah, I was looking for another one. Brain Atlas, if we go down here, blah, 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 find out more. Sure. We'll see if this is what I want. It is not. You can see that there's some explorative things in there. Multi-level human brain atlas, eBrain. Somewhere in here, I was able to look at the keeps asking me. Look at expression profiles for our samples. But anyway, the long story short, I wasn't able to get it to work out in a way that I thought would be appropriate to show. So if I ever get that kind of working out, then I will show it here. So that's the semi-supervised method. You need to have your profiles, which is your cell type. So a specific cell type. So if we're looking at immune cells, for example, we would have a fibroblast column, and then we would have all of the row names as the cell. They might actually have something here, or the row names as the gene expression. Here we go, or as the genes, and then your expression kind of falls in there. But today, what I actually thought would be nice to do is, if you don't have a single cell profile, which perhaps you don't, if you're doing this and you're kind of new to single cell and single cell transcriptomics as a whole, you would have no expression profile. So they have this unsupervised way. So remember last time when we did our clustering, we ended up with 14 clusters for our Umap here using the Louvain method. And if we were to import our data, so this is the same, this data here is what we had ended on last time after we ran the Louvain clustering and then did our Umap on all of that. Oh, here comes Puppy. What we need in order to start clustering this to identify the different cell types that we have with InSituType is we need our counts, and that's going to be in our RNA slot. So if we remember when we were first exploring our data, there were 4 different things that were in our object slots. We had, it's not happy. It is, we now have 4 because we reran our normalization, but we have our false code, RNA, which is actual cells, and then the expression profiles, and then our negative probes, which are kind of our false positives. And now we have our SCT. But when we run this, we'll actually use the unadjusted or the untransformed expression for our cells. So you can see that these are whole counts. They're not normalized in any way. So what we need to do is pull out our raw counts, which we can do by doing object assays. So we're going into that assay slot, looking at RNA, and then pulling out counts. Now this is a really big data set, so it's 188,000 by 6,000, remember. So we have our RNA, number of cells by our gene expression. So when we unpack this, it gets rather large, so 9.5 gigabytes. We just have to keep that in mind. Our negative is much smaller. So there's, I think there was 20? Yeah, 20 negative probes by all of our cells. So these are the false positives. We won't use false code, but the way that they described it on the website, perhaps in their new 6,000 plex, the false codes might be something that we can input into the data as well, or input into the InSituType function to help inform our clustering of our cells. Now once we have those 2 pulled out, so these are just matrices now, so count, large matrix, neg, it's a large matrix. We can get the mean for our rows. So if we do quickly neg, and we just look at the first 10 rows and the first 10 columns, we see that each row is a cell. So what we can do is we can average the negative probes for each row, and then we can look at that. So we have 20 total. So if we look at the first cell, actually, let's go back up. Let's look at 1 through 20. So these are all of them. We have 1, and then there should be another one, 2. Yep, so 2 out of 20, so that gives us 0.1 is our average, right? All right, so now we have our negatives, our average negative. Something that InSituType is, it's nice that it has, is that if we remember the first time that we looked at our object, perhaps we can look at our metadata. So let's do view object at metadata. We have a few columns where they did immunofluorescence, right? So we have histone, rRNA, and these potentially are not that informative when it comes to cell types. I'm not 100% sure what G is. Histone might be something where there's going to be more histones if the cell is actively multiplying versus just kind of hanging out there. GFAP, DAPI for the nucleus. But what we can do is we can use these in data to inform our deconvolution, as it were, using InSituType. So if we pull those out, now we have just our columns that are immunofluorescence for all of our cells. I had also removed DAPI just because it might be different depending on the cell types that you're looking at, but a nucleus should, naively saying, be the same size for the cells that are in a very small region. If we go back here, then what we can do is look at the average negative counts for all of our cells. So if we do head and then our negatives here, get our background. So what this is also going to help us with is some information that we can pass into our InSituType function to help inform our deconvolution with the Bayesian approach. So coming back to the immunofluorescence, what we can do is we can use that to identify some cohorts, is what they call it. So using all of these different columns here, we can split the data up into groups of more or less similar immunofluorescence abundances. If we look at table, this is not great. There's 50 groups. And it might be something if you're looking at a really busy tissue, you might end up with that many cell types. Another thing is too that all of our samples, I'm not 100% sure where they all came from, if they were picked specifically by a pathologist to be the same, but remember there's a lot of samples too. So depending on where all of our samples come from, this might be one type of tissue, 1 through 17, 18 through, I don't know, 27 might be another type of tissue, and then 28 through 50 might be another tissue type. We just don't know where exactly they all came from. Even if we look at the metadata, I don't believe this is awfully informative. Other than human brain, frontal cortex, we can actually look, let's see here. If we do unique object metadata $ tissue. Yeah, it's all the human brain. I don't think there's really anything else in here, I might miss it, that helps us identify that there are different tissues. But nevertheless, what that cohort will do is kind of help. So we can pass that all in now. So now we have our counts that we had taken out of our Seurat object as counts. We have the mean negative probe count for each of our cells. So remember, it's 0.1, 0, 0.1, so on and so forth. I can't remember. I might have scrolled way past it. Yeah, 0, 0.5, 0.2. We'll pass that in. We'll also pass in our cohorts that we got from our immunofluorescence. And then we don't have to, but since we were able to calculate our background for each of our cells, then we can pass that in as well. And it's just all going to help identify the different clusters that our samples make up right over here. Nclusts. So what this is going to do is it's going to have InSituType search through 10 clusters, 11 clusters, so on and so forth, all the way until it gets to 20 clusters, and then it will decide the best one. I picked 10 through 20 in this case because when we were doing low vein clustering, we ended up with 14. So we have 0 indexed and then 13 max. So 1 through 13, 13 plus 0 is 14. Just a happy middle here, 10 through 20. And then InSituType will go through and then pick what it thinks is the best fit for our data, the number of clusters. This takes a while. It doesn't take as long as one would think, deconvoluting all of that, but it does take some time. And if we look at the output here, we have our unsupervised clustering object, and then we have some slots. So we have clusters, so this is actually going to be the cluster that InSituType has assigned to each cell. So we can see here, first cell is cluster B, second cell is cluster D, so on and so forth. Then we have the probability, and I believe this is the posterior probability. So the higher, the better, is how I understand it. 100% that it belongs, that this cell belongs to B, and we'll see a flight path plot of that. Then we have our new profiles, log likelihood, so on and so forth. But the one that we can use are those profiles, probability, and then the clusters. So let's look at a heat map of our new clusters here. And it might take a while. There's a lot of cells, but what we'll see is along the y-axis is all of our cells. Along the x-axis is our different clusters. And this big block in here, if I had to wager why that's pretty empty, is because there's a lot of the custom probes that they have in here. So when you run InSituType, we can just grep. So let's do blah, 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 object at assays $RNA at counts. Let's do call names just to see if we get the right one. No, we need row names. We can actually pull all this back. So we'll do row names. Great, and then let's grep custom with a capital C, custom. We'll pull it from there. We want values, true, and then we want, we'll keep it. We don't want to invert, but then we want unique, just in case. Yeah, so they have, I don't know, 200, looks like 200 custom probes that are added onto here, so you can have potentially more resolution for a particular gene aside from what a nanostring has provided. Or, since there are only 6,000, you can have probes for something else. So for example, if they don't have, they should, but like TP53, you'd be able to add a probe for TP53 and get that resolution for some malignant cell population in your samples. But yep, let's make sure. I believe it's in the background now. There we go. So we can see that there's this huge block for N. Well, it's kind of interesting. We'll be able to see where that is when we do the labeling on top of a flight path, or we can even do a dim plot like we did last time to plot our Louvain clusters on top of our Umap. We have some pretty distinct signatures for A through I. They look much more muted. D's got a hot block down here. G's got a hot block up here. Just as an example. And then what we can do is we can actually plot those on top of our Umap, or as our Umap, right? So here I just have some colors that I picked up from one of their help pages, and then we're just plotting the Umap embeddings for our different clusters. So unsupervised clustering and then clusters, we could just use those for our colors. And here we have, remember N was the one that had that really hot block. And it looks like N is mostly over here. Maybe that's B. It's really hard to tell with so many colors. We have K up here. L's kind of in and amongst all this. A, some deep purple for C. And remember that this is only a two-dimensional representation of our Umap, which used 50 PCs. Maybe it was 30 PCs of the principal components we have 50 of. But now we have our InSituType assignments for our clusters on top of our Umap. So to view the confidence that we have, remember before I mentioned that we have those posterior probabilities for our different cells. So here this one looks really, oh, I might be looking at the wrong thing. I think I'm looking at the wrong thing. What we want is this one. Yeah, those are profiles. We have high confidence, B, D, so on and so forth. So we want closer to 1, and we can view that if we were to plot the flight path plot. We will just pass in our unsupervised clustering because that's what came out of InSituType's function. So if we go back up here, InSituType, we have our unsupervised clustering. And then the colors, we can just pass in the exact same cluster colors that we had for our Umap. And when we go ahead and plot that, this might take a while too since there's so many cells. we'll be able to see sort of the points that are falling between two centers of cluster assignments. So the closer that a cell is to 1 for the posterior probability, the closer that it's going to be to the center of the cluster. Let me see if I can zoom in here. And then if InSituType is unsure whether or not it belongs to cluster B or cluster A, it'll put it someplace in the middle of the two. So A is over here. This is also a two-dimensional representation, but we can see that D and B, there's this trail path between the two. So as it gets closer and closer to the center, the confidence or the assuredness that the cell belongs to cluster B drops, and at some point then it will flip to being part of cluster D. We can see that there's some of them that have very, very few cells assigned to them. So N, maybe 2 cells. There's one behind the label there. Maybe, I don't know, 10 if there's some that are hiding back behind as well. But there's lower confidence there as well. K is 1. We just want high values here, and this looks pretty good. If you have a lot of webbing between all of the points or all of the cluster centers, it might be worth digging a little bit further into why that's happening. Perhaps there's some probes that are failing, and for whatever reason, it's just not allowing InSituType to identify differences between cell clusters, or you have not provided a good range for InSituType to search within. So I believe if we count up the number of clusters that we have, we actually end up, in not telling InSituType about it, we actually end up with 14. I need to confirm that to be sure. So unique, let's check this out. Look at this. Then we need to do length. And there we go. So InSituType was able to identify the same number of clusters that low-vein clustering had picked up, and from basically just the gene expression itself. And this is not the transformed gene expression. This is the raw counts as well. Finally, what we can do is we can go back to that cell the field of view that we looked at the first time, right? So we looked at slide 1, field of view 1. So if we go ahead and run this bit, we'll be able to compare our annotations with the annotations, essentially classes, that InSituType gave us versus the first time that we were looking at the assignments provided by NanoString in their Surrat object. As we can see up here, most of the purple is here, more blue is scattered on this side. Let me zoom in. Yeah, so we can see blue is basically in the bottom right, and purple is in the top right. So let's go here and see. We have blue in the top right, blue and pink. So K and potentially L. And then we have orange and green, it's seemingly in the bottom right hand. There's some purple down here too. But without knowing the tissue, I'm not 100% sure what to expect. It looks like there's also green down here. But we are getting that separation, right? So they provided labels that put a cell type upper right and bottom left, upper left and bottom right, and we're also seeing the upper left and bottom right for our clusters as well. So I will put a link down in the description to the InSituType vignette page, and then I will also link to the GitHub repo for the script. I'll upload that to the Spatial Transcriptomics repo. That way we have that. And one last thing here, let's make sure that we save this, because last time I forgot to say that and I had to rerun it after closing. Save object. Next time when we come back, if I'm able to get the phenotyping to work, then I will show that one. Otherwise, what we can do is run differential gene expression from all these different clusters and then go into the human protein atlas and look at, okay, blue is showing a much higher expression of this gene. Okay, what kind of cell is that associated with? And we can sort of curate the cell types knowing that these are coming from the brain. We're not going to look for skeletal muscle as a cell type. But what we can do, since we do now have our InSituType assignments into our Surrat object, we can save our Surrat object as InSituType labeled. So we can go data, and then human frontal cortex, and then InSituType. We'll just call it that. We'll go ahead and save that. So if you guys thought this was helpful and are interested in these videos, please just let me know, and I'll keep making them. I understand that they get extremely long and sometimes well-winded, but I hope they're helpful to everybody. Bye.